Hi, I'm the great builder. Nobody knew me until my good friend, Mr. Hook, spotted me through a microscope and gave me a name. Living organisms know me better as cell. Does that ring a bell? You'll soon know more about me.
Every living thing is made up of cells. Your skin, blood, heart, every tissue and organ in your body begins its existence as a single cell that divides and divides again. The membrane of the cell is a bumpy film of fatty molecules. Proteins rise like trees from this oily layer to pump and channel minerals and nutrients through the membrane. Other proteins use their branches to trap hormones that activate the cell. Some hormones are small enough to slip through the membrane to instruct the cell to move, grow, divide, or even die. Larger hormones dock at the protein receptors. The receptors send messenger molecules to the enzymes and organelles crowding the cell. Some messages reach a network of tubes called the endoplasmic reticulum. Channels within the tubes then release calcium, triggering cell activity. Together, messenger molecules and calcium command microfilaments to contract or relax. They may also tell the cell membrane to recycle its components. Tiny honeycombs of proteins swallow up pieces of membrane along with hormones and their receptors. After ingesting its host, the protein structure falls apart, liberating a balloon-like vesicle into the traffic of the cell. The protein building blocks are then ready for a new round of ingestion. Mitochondria are organelles that provide the energy the cell needs to operate its pumps, motors, and micromachines. This miniature power plant uses motor proteins to cruise along microtubules on its journey through the cell. A thick soup of enzymes, micromachines, and organelles, called the cytoplasm, performs the daily tasks of the cell. The chore of making proteins for export to other cells is carried out in the tubular maze of the endoplasmic reticulum. Our small hormones plunge through the cytoplasm toward the cell nucleus. Increasing numbers of microtubules signal the presence of the centrosome. The centrosome sends out microtubules that lengthen and shorten in all directions, controlling traffic within the cell. The nucleus is the largest organelle of the cell. Guarding entry are the nuclear pores that sort incoming messages, routing the hormones toward the genes. Here we encounter the precious genes, the carriers of heredity. Genes are long, thin strands of DNA wrapped around bead-like proteins. Within a single nucleus, the DNA necklace can be made of 100,000 different genes. A hormone, before it can find its target gene, binds with a nuclear protein receptor. This couple then joins with other couples on the crowded dance floor. Together they attach to the particular gene the hormone came to meet. The gene then relays the hormone's desire for the cell to grow, divide, or die. To carry out the order, proteins gather at the site, forming a micromachine that reads and transcribes the gene. The genetic code is read on the DNA double helix. The twisted strands of DNA consist of complementary pairs of 4 nucleotides, the building blocks of the genetic code. We can think of the nucleotide molecules as letters of an alphabet and the genes as books. DNA is a giant molecule made of atoms of carbon, hydrogen, and other elements neatly organized into a double helix. As the micromachines twirl along the strand of DNA, they decipher the genes and transcribe them in the form of RNA molecules. Once completed, the RNA detaches from the DNA strand. The RNA then leaves the nucleus through the nuclear pore for the cytoplasm. There, the RNA will provide the code to make the proteins that carry out the hormone's instructions. If the order is to divide, the cell will make an exact replica of its DNA to pass on to the daughter cell. The process of DNA replication takes place in the nucleus, where micromachines make accurate copies of the original. Specialized enzymes within the micromachines replicate the DNA at many sites along the strand. In preparation for division, the identical meter-long DNA strands condense. Enzymes and structural proteins engineer a sequence of packing, folding, and coiling. They supercoil into micron-sized packs of genetic information called chromosomes. Many other things are happening inside the cell at this time. The centrosome has replicated, and microtubules grow in all directions. Mitosis, the most dramatic event of cell division, has begun. Now, pairs of sister chromosomes and a spindle built of arching microtubules occupy the cell's center. At first, the sister chromosomes glide along these microtubules. Located midway along each chromosome is a disc-like anchorage called the kinetochore. It is at this site that the microtubules fasten to the chromosome. The kinetochore on each sister chromosome is captured by a microtubule from the opposite pole. The chromosome pairs oscillate between the two poles. This astonishing tug-of-war is orchestrated by an army of motor proteins and enzymes. Finally, the sister chromosomes are aligned as couples between the two poles. Pulled by the microtubules connected to the poles, the chromosomes suddenly split apart. The function of mitosis, to produce and separate two sets of chromosomes and centrosomes, has been accomplished. Now, a contracting ring pinches the cell across its middle, forming two identical daughter cells. Daughter cells will grow and divide in turn. This is the amazing cell cycle, the basis of all life.
The cell prepares for meiosis just as it does for mitosis. But as the DNA condenses, each chromosome finds its homologous counterpart. These matching pairs of chromosomes hold equivalent genetic information, one set from mother and one from father. Next comes the first shuffle of nature's genetic deck. The two homologs trade genes in a process called crossing over. This forms new hybrid chromosomes. The spindles then place maternal and paternal chromosomes randomly on each side of midline. It is another shuffle that can lead to a host of different genetic outcomes. Finally, spindle fibers pull the homologs apart. This leaves the sister chromatids intact. With twice the amount of genetic information the cells will need, nature must cut the genetic deck in two. This second division produces 4 unique sets of chromosomes. Because they carry half the genetic information, we call these haploid cells. Many of them will mature into sperm and eggs, the reproductive cells called gametes.
do you turn that bite of food into a chemical that a cell can recognize and use as energy? The first step is altering the food into its component chemical compounds and then getting those molecules into your cells. That process is called digestion. Once inside your cells, the process of turning that bite of food into useful energy by cellular respiration begins. The process of digestion results with carbohydrates and other molecules being removed from the consumed food and transported into the bloodstream. From there, nutrients, like the carbohydrate glucose, will leave the bloodstream through a capillary wall and enter a tissue cell. Once inside the cell, cellular respiration will completely oxidize the glucose molecule, releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells. Cellular respiration is a four-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of two distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, two ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the two 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistance. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins. The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain. The electron transport chain is a series of membrane-bound carriers in the mitochondria that pass electrons from one to another. As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecules. Proteins in the chain pump hydrogen ions across a membrane. When the hydrogen ions flow back across the membrane through an ATP synthase complex, ATP is synthesized by the enzyme ATP synthase. Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water, a byproduct of the electron transport chain. All the high-energy electron carriers from the previous stages of cellular respiration bring their electrons into the chain. From this, the bulk of ATP from the entirety of cellular respiration is produced, a net of 32 to 36 ATP. In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP, the energy battery of the cell. On average, 36 ATP molecules are produced per glucose molecule that entered the cell. In the process of producing ATP, oxygen is brought in from the bloodstream to be the final electron acceptor in the electron transport chain, and the carbon dioxide that is produced as a byproduct is released. The goal of cellular respiration is to transfer the energy from the food that we eat daily into ATP that our bodies can use. This process starts with the eating of a snack or meal and ends with capturing the energy from the complete breakdown of the nutrients into energy and carbon dioxide.
If you want to know how a plant grows, it takes water, air and sunlight and makes cellulose. Every plant can do this fundamental process, and we can call this photosynthesis. Unlike me and you, plants need CO2, and they make oxygen that stops us turning blue. Every plant can do this fundamental process, and we can call this photosynthesis. It's a miracle how all the chlorophyll captures sunlight in the leaves of the plants and the trees. A plant of any size can do it if it tries, but we're not green, so we can't photosynthesize. Every plant can do this fundamental process, and we can call this photosynthesis. It's a miracle how all the chlorophyll captures sunlight in the leaves of the plants and the trees. Every plant can do this fundamental process, and we can call this photosynthesis. Every plant can do this fundamental process, and we can call this photosynthesis. Photosynthesis.